Hello, so my name is Konstantin Popadin, and today I will guide you through my poster. So let me share screen and start. So my poster is called A Billion-Year Trend of Amino Acid Substitutions in the Mitochondrial Genome. So another title is more functional. It is strongly asymmetrical mutagenesis of mtDNA shapes universal trend of amino acid gain and loss. So the graphical part of my poster is represented here, and now I will guide it to this uh, graphical part. And please feel free to contact me by email or Twitter. It has been shown that many amino acid substitutions are disappearing during the long-term evolution, such as alanine and proline, for example. We will call them losers. And other amino acids, they increase in frequency, such as cysteine and methionine. We will call them gainers. So this paper described this global trend analyzing nuclear genomes of eukaryotic organisms and bacteria. But till now, it's unclear what is the main driving force for this gain and loss of amino acid. Is it selection or mutagenesis? In my project, we decided to concentrate on mitochondrial genome because in this case, we know rules of mutagenesis. And so we can predict what kind of amino acid substitutions we expect if mutation spectrum is more important as compared to selection. So let me explain a little bit about mitochondrial genome. So mitochondrial genome is circular and it has asynchronous replication. The main idea is that during replication, one strand, which is called heavy strand, is single-stranded for a long period of time. And exactly this mode, so single-stranded condition, is very mutagenic. And two, the most common transitions, C to T and A to G, they happen predominantly on this heavy chain. That's why if we reconstruct mutational spectrum, so probabilities of substitution from one nucleotide to another in the most neutral positions, so in this case we calculated mutational spectrum for synonymous substitutions, we see that majority of mutations are C to T, this type of substitution, or A to G. This strong mutational bias is asymmetrical. For example, C to T, which happens on heavy chain, would be equivalent to G to A, which can happen on opposite strand, which is called light strand in case of mitochondria. But C to T on heavy chain is 10 times higher as compared to G to A on heavy chain. So mutation spectrum is very asymmetrical, and thus it will shift nucleotide content, codon usage, and maybe even amino acid usage. So that's what we want to prove. Our question, what are the consequences of this asymmetrical mutagenesis? So once more, equivalent substitutions, for example, C to T and G to A, they are not similar. And another pair of equivalent substitutions, A to G and T to C, they are also not balanced. So let's have a look on mitochondrial genetic code. So the most common substitutions which I described, C to T on heavy chain and A to G on heavy chain, we can convert them on light chain because light chain is a reference sequence. And in all databases, we use a light chain. So we can see that these two substitutions, they really shape codon usage in human mitochondrial genome. Here, numbers describe frequency of different codons. And for example, if in case of alanine, we can see that GCT is more rare as compared to GCC because many mutations, they go from T to C. So from GCT to GCC. And we can see all these horizontal stripes, meaning that in all cases, in all amino acids, codon which ends on C is more common as compared to codon which ends on T. And this is a result of mutagenesis. And the same situation for a second transition from G to A. So GCG is more rare as compared to GC8 because of substitution from G to A. Okay, so it has been shown that human synonymous codon usage is mainly shaped by this strong and asymmetric mutational bias. And now we would like to ask, what about amino acid substitutions? Maybe even amino acid substitution can be affected by this strong mutational spectrum. So let's have a look on the same code, but now we take into account not just changes in synonymous codon usage, but between amino acids. So let's again repeat two the most common substitutions from T to C and from G to A. So it will create this pattern. Here we have all codons which has G in the middle. So if we have G to A, so the fourth column will mutate to the third one because of mutation G to A. The same mutation T to C. The first column, because T is in the middle, will mutate to the second column because of C in the middle. Okay, and the same here from T to C, from G to A. So these two mutations, two the most common transitions, will create this pattern that some amino acids are expected to disappear, like cysteine, phenylalanine, and valine. We will call them expected losers. And other amino acids, they expect to increase in frequency, proline, histidine, lysine, threonine, etc. And some other amino acids, let's call them intermediate. So we don't have strong prediction about frequency of these amino acids. So this hypothesis that mutation spectrum can shape amino acid substitution is testable. First of all, we expect 27 pairwise strands. For example, from valine to alanine should be more common substitution as compared to backward. So we have expected gainers in the middle. We have expected losers at the corners of genetic code. Also, we have a majority of genes which are coded on heavy chain. So mRNA of all these genes will be equivalent to light chain, reference chain, which we discuss now. But one gene, ND6, is coded on light chain. So its RNA will be identical to heavy chain, so opposite. That's why we have one gene where all these arrows will be completely opposite, so we can use it as a proof of principle. So let's go for empirical data. And I will describe in details one example coming from somatic mutations in mitochondrial genome in human cancers. Let's take into account our substitution from valine to alanine. So among all somatic mutations in human cancers, we found 78 non-synonymous substitutions from valine to alanine. And now we need to normalize it on number of valine. So finally, with 78, we divide by 136, and we have 0.5 or 0.6. So this is rate of substitutions from valine to alanine. This is rate of expected substitution. Now, let's have a look. What about opposite trend? So we found 27 from alanine to valine, sorry. So 27 from alanine to valine. And totally, we have 245 alanine in mitochondrial genome of human. So again, we divide 27 by 245, and we have this 0.1. So this is rate of substitution from alanine to valine, so which is not expected. So we have 0.6, which is a rate of expected substitutions, and 0.1, which is rate of unexpected substitutions. We can divide expected on unexpected, and we take a log ratio, and we have 2.3. So 2.3 is higher than expected 0. So 0 is our null hypothesis. And now I explain just from valine to alanine, but we have many amino acid substitutions, and red color marks majority of genes which are coded on heavy chain. And the RNA of all these genes is equivalent to light chain. And in case of ND6, I don't want to explain details, but indeed all ratios are negative, so they are opposite. So now we can just visualize all these ratios as a histogram. So we expect 0, it's our null hypothesis, but for majority of pairwise substitutions, we have a ratio more than 0. And for ND6, all of them are less than 0, as expected from our hypothesis. Next, we prove the same trend using not only human cancers, but we use human polymorphisms, human pathogenic mutations, polymorphic data from vertebrates, and in all cases, we found quantitatively similar trends. Every time, rate from losers to gainers is a bit higher as compared to rate from gainers to losers, in line with our hypothesis. Next, we decided to count fraction of losers and fraction of gainers for many complete genomes. So we took into account reference genomes, complete mitochondrial genomes for fungi, plants, invertebrates, and vertebrates. And we calculated fraction of losers and fraction of gainers in all mitochondrial protein coding genes. And we can see that gainers, red, they are more common, at least in vertebrates, as compared to fraction of losers. And after we decided to come back to the past and see what was ancestral mitochondrial genome. We know that mitochondria originated from alpha proteobacteria. So we found orthologs of mitochondrial protein coding genes in bacterial genomes, and we estimated fraction of losers and gainers in alpha proteobacteria. And interestingly, we see that initially a fraction of losers was high enough, let's say 30%, but fraction of gainers was low, 20%. And at some moment after origin of mitochondrial genome, gainers started to increase in frequency, but losers started to decrease. So there are two explanations for this global trend, which are non-mutually exclusive. When we move from bacteria, fungi, plants, invertebrates, vertebrates, from left to right, we decrease effective population size. So the highest effective population size in case of bacteria, intermediate in case of fungi, plants, and vertebrates, and the smallest effective population size in case of vertebrates. So if there is mutational bias from losers to gainers, and this mutational bias leads to many slightly deleterious substitutions, all these slightly deleterious substitutions can have higher chances to be fixed in organisms with low effective population size. So that's why losers are really decreasing and gainers increasing from high effective population size bacteria to low effective population size vertebrate species. So this is our favorite explanation. On top of this, we can suspect that in case of vertebrates, and especially in case of warm-blooded vertebrates, not only mutational bias is strong, but also just mutational rate is strong. That's why all these species, they come to equilibrium, they accumulate mutations faster. So finally, we observe that mitochondrial mutation spectrum creates a very strong shift in amino acid composition. And comparing with alpha proteobacteria, we observe strong difference in fractions of losers and fractions of gainers. So we conclude that modern composition of mitochondrial genome of all vertebrates is significantly shaped by mutational process. And mitochondrial genome is contaminated by huge amount of slightly deleterious mutations which were accumulated during billion years of evolution. Thank you for attention. So I guided you through this poster. Thanks to all co-authors. Please feel free to write emails to me. And if you're interested in our topic, I would like to advertise two bioarchives from my group. One bioarchive is entitled Mammalian Mitochondrial Mutation Spectrum as a hallmark of cellular and organismal aging. And the second, that mitochondrial mutation signature of temperature in ectothermic and endothermic vertebrates. So in these studies, we demonstrated that mitochondrial mutation spectrum is sensitive to longevity of different species and to temperature of environment in case of ectothermic animals. Thank you. I will stop share and looking forward for your questions in Slack, email, and Twitter. Goodbye.